Assalamualaikum, I'm Anila from Government College University, Faisalabad. I am a student of Zoology. Today's topic of my presentation is evaluation of antimicrobial properties in fish skin, mucus extracts of major carps. Fish, diverse group of animals. First of all, I will speak about introduction. Fish is a diverse group of animals, highly specialized for their aquatic existence and comprising almost half of the number of vertebrate species. Globally, fish provides about 16% of annual protein. Global fish production was 171 million tons in 2016. Pakistan fisheries contributes about 1% to its GDP. Per capita fish consumption was about 1.9 kilograms annually. Fish coexist with many of pathogenic and non-pathogenic microorganisms and have complex defense systems which ensure fish survival. Infectious diseases cause multi-million dollar loss annually and reduce the economic income. It causes mortality in fish. Innate defense system helps a fish to maintain healthy state under normal conditions. Skin mucus, an important component of innate immune system, and act as a barrier between fish and the external environment and plays a frontier role in fish protection. Continuously get replaced and prevent colonization of bacteria and fungi. It also contains a number of factors of innate immunity like proteins, enzymes such as lysozymes, immunoglobulins, complement proteins, lectins, C-reactive proteins, phosphatic enzymes, alkaline phosphatase, and other components of antibacterial proteins and peptides. Fish skin mucus exhibits significant and specific antimicrobial, antifungal, and cytotoxic activity. Composition and rate of mucus secretions varies from species to species and in response to microbial exposure, environmental fluctuations, etc. Fish plays an important role in respiration, ionic and osmotic regulation, reproduction, excretion, disease resistance, communication, and protection, potentially make mucus a highly multifunctional material. Let's move towards materials and methods. First of all, experimental fish, Labeo, rohita, Cirrihinus, and Catla were purchased from government fish hatchery, Kiana Road, Faisalabad, 250 to 300 grams in weight. Fish was stocked in glass aquariums having water capacity of 36 liters. Then fish were acclimatized to the laboratory conditions for 15 days, fed on 32% basic protein diet. After acclimatization for 10 days, fish were used for the mucus collection. Before mucus collection, fish was starved for 24 hours and was placed on a specimen tray. Mucus secreted on the surface of body was collected as a sample with the help of sterilized spatula and was stored in sterilized apron drums. Then mucus was centrifuged at 5,000 rpm for 15 minutes and stored at 4 degrees centigrade. Then acidic mucus extract was prepared. For acidic mucus extraction, 1 ml of fish mucus was mixed with 0.5 ml of 3% acetic acid, which obtained concentration of 2:1. 2% was fish mucus and 1% was 3% acetic acid. Then apple drops were placed in boiling water bath for 5 minutes and then was cooled in ice and centrifuged at 12,000 rpm for 35 minutes. Antimicrobial assay, while the previous method was used to determine the growth of inhibition effect of skin mucus against the bacteria and fungal strains. Then let's move towards the results. Zone of inhibition of gram-negative bacteria, Aeromonas, hydrophila against Aeromonas, hydrophila by Labeo, Cirrihinus, Catla, and control was Staphylococcus antibacterial drug. The highest zone of inhibition was observed by the Labeo, which was 20 mm in diameter, and Cirrihinus comes at second position, 16 mm zone of inhibition, and Catla with 13 mm diameter of the zone of inhibition, and the control drug with 25 mm diameter of the zone of inhibition. Then at second number, gram-negative bacteria Staphylococcus aureus, the zone of the graph representing the zone of inhibition of all the fish mucus extracts, Labeo, Cirrihinus, Catla, and control drug. In the against the Staphylococcus aureus, highest antimicrobial activity was observed by the Labeo, then Cirrihinus, and Catla. The control drug shows the zone of inhibition of 25 mm, Labeo, 23 mm, and Cirrihinus mirrigala and the Catla with the diameter of one inhibition of 21 mm in diameter. Then zone of inhibition, antifungal activity of three Carps species against the Fusarium axis of spores. The highest antimicrobial activity was shown by the Cirrihinus mirrigala with the with diameter of the zone of inhibition 19 mm. Then Labeo and least activity was shown by the Catla. Zone of inhibition of fungal species as Aspergillus niger by Labeo Cirrihinus mirrigala and Catla. The highest antifungal activity was shown by the Labeo which was 17 mm in diameter and 15 mm in diameter of the zone of inhibition was by the Cirrihinus mirrigala and 13 mm diameter of the zone of inhibition was Catla, and 25 mm diameter of the zone of inhibition was the control drug which was gentamicin as an antifungal drug. This graph shows the comparative analysis of all the fish mucus extracts against all the tested bacterial and fungal strains. Blue graphs are bars are shown by the aureus, pink, green, and each color representing a different bacterial or fungal strain. And fish species. As the graph representing the highest antimicrobial activity was represented by the Labeo against the aureus, which was 23 mm in diameter. And the control and at right controls are also representing the diameter of zone of inhibition. The conclusion, the findings of the present experimental trial suggested that the acidic mucus extract of all fish exhibited the antimicrobial properties against the selected bacterial and fungal strains, which might play an important role in fish defense against the pathogens. Thus, it appears to the possible use of steam mucus of carps as a potential source of antimicrobial agents in alternative to antibiotics against the fish and might be in human infections. References and thanks for your attention.